With respect to Dr. Cordova, Antoinette is a field application specialist at EMIT Imaging, and Dr. Cordova is an expert in high-resolution 3D fluorescent imaging and drug discovery, with work spanning from many clinical applications from oncology through to neurology. Previously, Dr. Cordova was part of the Georgetown University Medical Center for Drug Discovery, where she led projects in tissue-specific drug delivery systems for breast cancer and addiction, in addition to peptide-based therapies for Alzheimer's disease. Dr. Cordova is a medicinal and inorganic chemist, molecular biologist, and imaging expert. She received her PhD from Georgetown. And before I forget, my name is Steve Chakowitz, and I'm the Chief Commercial Officer for EMIT Imaging. Started recently, about a year and a half ago. It's been very exciting. We've been making a lot of progress. So again, thank you very much. We're going to have questions at the end after all three speakers have gone, so save your questions for that point. Okay, thank you very much. Antoinette?
All right. So as Steve mentioned, hello, my name is Dr. Antoinette Cordova. I'm a field application scientist at EMIT Imaging. And today I will be speaking about Xerra. You will meet Xerra, and I hope by the end of my talk, you'll be able to understand how you can maximize data collection from preclinical models. So let's begin. So to give you an overview, I will be discussing what is cryofluorescence tomography, or CFT. It is the optical imaging modality that is featured on the Xerra. I will also highlight the research advantages of using CFT, in addition to going over the workflow and give you an idea how it can integrate into your existing imaging workflows, in addition to going over several preclinical applications that CFT technology and Xerra can be used for. So, what is cryofluorescence tomography? It is actually an imaging technique that captures 2D fluorescent and white light images. This is an ex vivo modality, so it's ex vivo serial sections, and we compile them into 3D images. As you can see on the far left-hand side, that is a cynomolgus head. We also can do whole mice, in addition to a rat brain or a mouse kidney, so we have a huge volumetric range that can be captured with our modality. So you might ask, why did CFT even come about? CFT actually fills the void across imaging modalities. As you can see from this graph, most are very familiar with cellular microscopy or MRI, CT SPECT, in vivo imaging modalities. But as you can see, CFT actually fills the void where there's orders of magnitude that can be addressed, where tissues can be interrogated and also overcome the limitations of in vivo imaging, for example, low sensitivity and depth penetration issues. And that is one of our 3D images. So now let's meet Xerra. Xerra is our cryofluorescence tomography platform. It features a 12 megapixel high resolution camera found in the tower. In addition to a 10 micron slicer, it also features 6 high-powered lasers ranging from 470 to 780 nanometers, in addition to 7 narrowband emission filters that are ranging from 500 to 850 nanometers. So you can have a whole host of different fluorophores or multiplexing using our platform. So in the end, you're able to achieve, obviously, 3D fluorescence imaging that is not only quantitative, but also reflects micro-resolution. In addition, the process is automated. A user will come up to the platform, use the touch screen, and then actually be able to set up their study design and leave. You can use a remote desktop application to just keep up to par with your study. And then by the end of the next day, you will see what you couldn't see before. So before that, I will also highlight some research advantages that I think will be very important for you as you can consider using CFT technology and Xerra in your imaging. So first is superior resolution. We provide down to 20 microns for detailed anatomy. We offer 5 different fields of view. In addition to high sensitivity, we're able to detect signal deep into the tissue. We're also high throughput. We can process up to 5 mice on our biggest block, and you will get 5 different data sets from that one block, so one experiment. We can also process multiple dissected organs on the same block, and our technology is also robust. So most of you, I'm sure, are doing some sort of in vivo imaging, CT, SPECT, MRI. we can actually co-register our data, combine it with CFT, in addition to tissue microscopy, and this is all from the same animal. We're also streamlined. Our technology does not require you to fix, perfuse, tissue clear, radiolabel, anything of that nature in order to maintain anatomical context. In addition, we're versatile. We offer a great range of visualization ranging from 470 to 780 nanometers. So now I'll go over the Xerra workflow. So how does it work? It's basically in 3 steps. First, the sample is prepped using OCT in the field of view of your choice. Then the sample is then placed into the Xerra as seen. And third is the automatic slicing that happens thousands of times based upon your model. So to just give you an overview, you begin by preserving or cryopreserving, embedding your model, whether it be whole mouse or, for example, small organs, whatever it is of your liking. And then we have a fully automated process where we have a high-resolution scan. We interrogate up to 10 microns of width of tissue. This will happen thousands of times depending on your sample. And in the end, you will have a 3D aggregate image. But in addition to that, we also offer 2D fluorescence and white light imaging that you can also just really deep dive and go into your data in addition to 3D imaging. What's also wonderful about the Xerra and just CFT technology as a whole is that it replaces months of tedious lab work, and it also reduces the use of animals because you'll have an additional endpoint that was not in your traditional workflow. So you can design experiments at the very end of your studies in order to capture, for example, like another dose-dependent study at the very end of your work. So some of the preclinical applications that the Xerra has actually been used for range, for example, from gene expression, drug discovery, getting PK, biodistribution studies, neuroscience, cancer biology, using understanding tumor microenvironments, using CFT technology, and in addition to immunology, understanding immune responses. There are so many ways that we can even assist you in your experiments and just optimizing your imaging with high-resolution imaging that we offer. So in summary, I hope that you've been able to grasp from this presentation that Xerra is a high-resolution imaging platform with a wide variety of preclinical applications. The CFT will build and function as a complement to in vivo imaging. In addition, it will overcome the low resolution and low sensitivity limitations of preclinical imaging. And lastly, and I think one of the most important parts for me personally as a scientist, is that you actually maximize the data you collect from one animal. And the fact that you're able to get a comprehensive data set that also includes anatomy, the white light, I think can just be such a valuable addition to your work. So with that, I just want to highlight that you can get more information, obviously, from emitimaging.com. You can reach to us via email, on LinkedIn. But also, we're at the booth. We're at booth 307. I'm more than happy to talk to you about your thoughts on the platform. In addition, if you're very much interested in designing a study with us, and we can just really go further. So I guess we will have questions at the end, but thank you so much for your time.